All right. Yeah, hello, my name is Julia Engelmann. I'm a tenure track scientist at the NIOZ, and I am a computational biologist by background, working together with a lot of marine researchers at the NIOZ, and some of these projects involve the interactions between plastic and the biological world. So the project that I want to show you today is about bacterial and eukaryotic microbial interactions on plastic particles that we looked at with causal network modeling. So plastics has been around since about the 1920s, with a steep increase in production since the 1950s, and still increasing today. A part of that plastic, unfortunately, ends up in the ocean with an estimation of about 10 million tons per year that enter the ocean coming from the land. This is, of course, very unfortunate, but also the plastic concentrations in the ocean vary depending on the ocean currents that lead to accumulation of plastic in certain regions of the ocean. And these 5 hotspots where plastics really accumulate have been termed garbage patches. And you might think of or see pictures in the media with plastic bags and bottles and these large pieces of packaging floating around. But that's actually not a very realistic picture of these garbage patches. Most of the particles are quite small, in the size of millimeters or even below. Now, in this model by Van Sibyl from a few years ago, it can be seen that the Mediterranean, although it's not a garbage patch, it actually had the highest number of plastic particles that can be found in the water there. So we were looking at data from the expedition MAT, Mediterranean in Danger, expeditions that took place in 2017 and 2018, where a total of 131 manta trawl net trawls were performed in different places of the Mediterranean coast to collect plastic particles in the size range of 1 to 5 millimeters. And a lot of volunteer work has been performed to collect these particles, and we're quite thankful for that. Now, from these particles, one particle was selected per sample as representative, and the biofilm taking off of these particles and DNA extracted, and then the bacterial V6 region of the 16S and the eukaryotic V4 region of the 18S was sequenced with amplicon sequencing. And then we clustered sequences using Swarm and Silver 132 as reference database for the bacterial, and Proteus ribosomal database as reference database for the eukaryotic sequences to generate O2 abundance tables, and use these then for downstream analysis, looking at profiles and networks. And this is just a very broad view. I don't want to go into detail here, but just such that you appreciate that there's quite some variation in the microbial community composition on these particles. So on the upper half of these bar plots, you see the bacterial community is classified in phyla and classes, and the lower half shows the eukaryotic supergroups. And yeah, each bar represents one sample. And yeah, you can see that even on this high level, there's quite some variation. Now, if we look at a non-metric multidimensional scaling plot of these samples, we can see that the marine samples, where I put this blue circle around, cluster together, and they cluster apart from a subset of the samples that were taken from rivers, so plastic particles from rivers and from ports, and also some seawater samples at the top that cluster together. So these samples have distinct communities. But we wanted to look into interactions, so let's zoom into this. Now, if you think about interactions, the first thing maybe that comes into our mind is to look at correlations. How are abundances of particular species or O2s correlated over time or over a series of samples, like we have in this study. And then, of course, with the thoughts in mind that correlation could be caused by either metabolic dependencies, symbiosis, competition, predator-prey, or because organisms share the same niche. Now, while correlation can give you a lot of insights already and hypotheses about these features, we also have seen that this gives a lot of false positive predictions. So if you want to be a bit more specific, then network approaches are a good way of reducing the number of predictions to more realistic ones. And that is by taking, for example, multiple features into account at the same time, like partial correlation, where you correct the correlation by the surrounding kind of features that you have in the data set. In this project, I was working with causal interaction networks, and this is asking a little bit of a different question than the co-occurrence networks. The causal networks specifically ask what happens to a feature if I perturb an upstream feature. And to being able to say that, I need to have a directed network because I need to be able to distinguish the cause from the effect. And it might sound a bit strange, but actually you can get directed networks from observational data, and I'll show you in a second how we do that. Now, these directions are something special, I would say, about these causal networks, enforcing sparsity to enrich for true interactions and reduce the number of co-occurrences that are due to other factors that you're not interested in, including spurious correlations, but also because organisms share needs. It's not something that is special to these kind of networks, but it also applies. So how do we get the directions into the networks? For this, we use conditional independence, and I'll show you some simulated data to exemplify the idea. So imagine we have a very small community that consists only of 3 members. And if we look at the abundance of these members, we observe that member A and member C are correlated in abundance. Let's say this correlation is significant, and therefore we draw an edge between the 2 features. If we look at the abundance of feature B and C, we also find that they are correlated. And again, we draw an edge to reflect that. Now, if we look at the correlation of A and B, we find that they are not correlated, and that can happen, of course. But then if we look a bit more closely and also take into account the abundance of C, so taking more than the 2 features in question into account, then we see that when C is low, A and B are actually anti-correlated. And I highlighted these dots or these samples where C is low with the blue crosses. So that's whenever C is low, and the black circles are still the samples where C is high. Now, if this is the case, if C carries information about the status of A and B, then we say that A and B are conditionally independent. So depending on the status of C, A and B are correlated or are not correlated. And to give you a real world example to exemplify this, think about a college that accepts students who are either really sporty or really smart. Now, if you know that a student has been accepted to the college, you know that that student is either sporty or smart, probably not both, because that's very unlikely to happen. And he must be at least one of that. So in this case, the information about C, admission to college, gives you information about the status A and B, being sporty or being very smart, being smart. While if students that have not been accepted to this college, their values of A and B, sportiness and smartness, are probably uncorrelated. Now we can use these data structures. Or actually, yeah, I didn't say that. Now that we know this information must be going from A, B to C, so smartness and sportiness comes before acceptance to college, we can make directed edges from A and B to C. And if we apply this to an observational data set, we can infer a partially directed graph, because these structures don't apply to all of the features that are in the data set. The graph will be partially directed. Here I put an example where all of the edges are directed, but that's not the normal case. It's just for illustration. So now the cool thing about causal effects is that if we do have the correct graph, the causal effect is identifiable. So if you have the correct graph that includes all the features that need to be there, you can identify the causal effect. Reality is unfortunately that we never have enough samples to get the correct graph, and usually there are also unobserved variables, and that will distort our inferred graph. Nonetheless, we can use the causal effect estimation to give us hypotheses about potential interactions and about potential causal effects. Now, to calculate the causal effect, what we actually do is that we ask the question, what happens to a specific feature, in this case, Y, if we manipulate the abundance of an upstream feature, let's say B3. And to calculate the causal effect, you basically do a regression of the feature B3 and all of the upstream features in the graph on your variable of interest, in this case, Y. And by doing this, you are correcting the causal effect for B3 on Y for anything that happens upstream of B3. So you really want to know the causal effect of B3 and not the causal effect of a eukaryote that is upstream, in this case, of this bacterium, and that bacterium is just mediating the effect. Now, when I do this to the Mediterranean data set, actually that's a lot larger. There are many swarms and many effects, and that's not very convenient to look at. But just to give you an impression that you can get causal effects for a number of causes and for a number of targets, and then use that data to dig into and look for hypotheses that you want to follow up with. And here are some hypotheses that we selected, and that could be followed up with experimental approaches, which are really needed to confirm the predictions from the network calculations. And I'm showing here just one example for the different for interactions between different and the same domain. So between the bacteria, one of the strongest effect from a bacterium on another bacterium was from a phylomicrobium O2, which is known to live on plastic surfaces, which had a positive effect on Flavobacteriaceae O2, which is an early colonizer of any surface in the ocean. So the hypothesis would be that phyllomicrobium is somehow helping the Flavobacteriaceae to colonize the plastic. From the bacteria on eukaryote effects, we found that, again, a phyllomicrobium O2 had positive effects on several diatoms, chlorophytes, and rhodophytes, which are photoautotrophs. And the hypothesis here would be that that bacterium is providing a cofactor, for example, vitamin B12, that these phototrophs need. For the eukaryote interactions, we saw a radiolarian Colosseum something with a positive effect on a Syndinialis dinoflagellate. And Syndinialis dinoflagellates are often parasites, and such that could be a parasite of a radiolarian host. And considering the eukaryotic bacteria interactions, we find a red algae, a rhodophyte, with quite a number of positive effects on several bacterial O2s. And here we speculate that bacteria represent the eukaryotic microbiome. That O2 hasn't been identified to species level, but the closest relative is this tsunami and Trans-Pacifica events. All right, so to sum it up, I hope I could show you that plastic particles harbor complex microbial communities with varying composition. And causal effect analysis proposed interactions within and between the bacterial and eukaryotic domains of life. We speculate that the eukaryotic microbiomes contribute to the plastisphere, so that's the microbiome on plastics interactions. And again, I want to stress that the hypothesis from causal effect analysis need to be experimentally validated with fish analysis at the least, or experiments to really prove that they are correct. And finally, I would like to thank you for your attention. Get in touch if you want to discuss or have more questions than we can address today. And I would like to thank co-authors, Linda, Alejandro, Tosca, Eric, all the NIOZ people that support and discuss with me, and of course the funders.